Sí, gracias.
Ok, gracias, Juan Carlos.
So now with the announcement, Adrian, we just wait a few seconds and we start. Juan Carlos, let us know when the YouTube broadcasting starts.
Ok.
Adelante, doctor.
Gracias, Juan Carlos. Good morning, everybody. Good afternoon, as maybe with Adrian and his colleagues in England, and good evening, even with our colleagues Sukumar in India. Thank you very much for joining to this last session of our virtual pre-Congress Symposium in order to encourage people to come next year to the International Conference on Mammoths and their relatives that we will host in Mexico City in September 2025. I thank you today, our presenter, Dr. Adrian Lister, and in a minute I will present. Also, here we have our co-host in this Seminario Relaciones, Hombre-Fauna, Eduardo Corona. So we are happy that we are happy today, probably is finishing with a very wonderful invitee that has been very supportive to always to our Mexican paleontology and now to our Congress, Dr. Adrian Lister. Dr. Adrian Lister is a research leader in paleontology, and certainly I will mention he's probably the most outstanding specialist in mammoths at the world. At the Natural History Museum in London, he obtained his BA and PhD in zoology from the University of Cambridge, and from 1991 taught at University College London, where he became professor of paleobiology, before moving to the Natural History Museum in 2007. His research interests are in the evolution of mammals from the ice ages to the present, on which he authored more than 200 scientific papers, while his 2 books on mammoths have sold over 60,000 copies in 6 languages. Other research interests include the dwarfing of mammals on islands, the causes of extinction of large mammals at the end of the Ice Age, and Darwin's fossil collections from the Beagle voyage, which formed the basis for his book, Darwin's Fossils, a really nice book, I should say. As well as his research on fossil mammals, Adrian Lister also studies the taxonomy of the living representatives and had led expeditions to study living elephants in Nepal, India, Borneo, and Ghana. He's a member of the Asian Elephant Specialist Group of IUCN and a frequent contributor and advisor to TV and radio programs on fossil and living mammals. And should mention that Adrian very kindly visited us several years ago to look at some of our deposits, and we hope he can be here next year. So before go ahead with Adrian presentation and no more delay, I just want to acknowledge our people that have during this presentation. So we want to acknowledge the Coordinación Nacional de Antropología, and particularly the Subdirección de Tecnologías de la Información, to Salvador Cristóbal Jesús Portilla Martínez, Luis Olaf Ortega Olmos, and Juan Carlos Carrillo, who is today behind that big red INAH symbol, who had made possible the transmission to the YouTube channel of the coordination. And we also hope, and we want to thank you to the head people from Centro, INAH, Morelos, and Subdirección de Laboratorios y Apoyo Académico, since they always help us to provide these spaces. And well, without any further delay, I introduce Dr. Adrian Lister. Thank you very much, Adrian. I'm sure we will enjoy this presentation.
Thank you very much, Joaquin. I will just share my screen for the slides. Do you see that okay?
Yes, it is.
Lovely.
Now ready.
Okay, well, I'm very pleased to be presenting the last in this wonderful series of talks as a preface to next year's conference. And I'm going to talk about the evolution of the mammoths. And I'll start off by looking at the morphological evidence, and then I will add what we have learned recently from the molecular So we can start off with a phylogeny of the elephants. This is one of the most recent trees that we have based on nuclear and mitochondrial DNA. And at the top there, you can see Mammuthus primigenius, the woolly mammoth, and not surprisingly, its sister group is the North American Columbian mammoth. And as long predicted from morphology, actually, the living Asian elephant, Elephas maximus, is the closest living relative of the mammoths. And the divergence date calculated from the molecular evidence of about 7 million years, in this instance, somewhat unusually, actually does fit pretty well with the fossil record, because the earliest representatives of both these genera in the fossil record in Africa is about 6 million years ago. So that works pretty well. So I'm going to show a series of maps, which I call a brief history of mammoths, essentially based on the fossil record. So those earliest mammoths from about 6 million years ago appear in southern and East Africa. And that's the species Mammuthus subplaniforms. And although this has been described as a wastebasket taxon, because probably some remains belonging to other taxa have been included within it, nonetheless, there is genuinely an early late Miocene mammoth. The actual type material of subplaniforms is genuinely the earliest known mammoths that we have. And there's a beautiful molar from the South African site of Langebaanweg, with its 8 or so plates in the last molar, very low crown, as you can see there. And then after that, into the Pliocene, we have a sort of small diversification of mammoths in Africa, producing Mammuthus africanus in North Africa, and some rather poorly known remains from East Africa, from Ethiopia, which are the Hadar form, still not with a species name. But mammoths were never particularly successful in Africa, actually, although that's where they originated. They were soon taken over, really, in terms of spread and numbers by the so-called Elephas reki complex. But about 3.5 million years ago, we first pick up mammoths north of Africa, and we have these remains, Mammuthus romanus, from Britain, from Italy, from Romania, 1 or 2 other places in Europe, and more recently discovered also in China. So that's the basis for that distribution, essentially in the temperate zone of Eurasia. Romanus, name based on its type material from Romania. And here is one of the better preserved specimens. This is a pallet from the Montopoli site in Italy, about 3.5 million years old. And these last molars, the Romanus species, generally have 9 or 10 plates in the last molar and a somewhat higher crown than the African forms. Some point up to about 2.5 million years ago, Romanus transitioned into the much better known species Mammuthus meridionalis, also known as the southern mammoth or the ancestral mammoth, which once again, it was named for Europe, is also found right across to China. And we don't really know the details of that transition because Romanus itself is very poorly known. Meridionalis, however, is much better known. And then subsequent to that, through the early to middle to late Pleistocene, we have this classic evolutionary sequence of species from Mammuthus meridionalis in the early Pleistocene, Trogontherii in the middle Pleistocene, and then Primigenius coming in the early Pleistocene. Those dates are based on European finds, and I draw attention to the earliest Trogontherii in Europe at about 0.8 million, and the earliest Primigenius at about 0.2 million. We'll come back to that later. And you can see how the teeth, especially in the stage from Meridionalis to Trogontherii, which I think is the most substantial evolutionary change, where we have a great multiplication in the number of plates and increase in the crown height, And then from Trogontherii to Primigenius, there's really not much more crown height interest, but some increase in plate count. And the classic story of the skull, which getting shorter and higher when you get into the Primigenius stage there. And then, so then we've got the Meridionalis. It was set up in Europe through the early Pleistocene, and then between about 1.5 and 1.2 million years ago, the first mammoth fossils appear in North America. And because Meridionalis was the only species originally known to occur in Eurasia at around that time, it was assumed that Meridionalis was the earliest mammoth that got into North America. That made sort of perfect sense. And that led to what I call the standard model of mammoth evolution, which I myself accepted in the earlier part of my studies, where Meridionalis is the ancestor which in Europe gave rise to Trogontherii and then to Primigenius, and then went over into North America, and there was a kind of parallel evolution from the so-called Mammuthus imperator to the well-known Mammuthus columbi of the late Pleistocene. But this story had to be revised, really, from the early 2000s, when remains referable to Mammuthus Trogontherii were found much earlier in Eastern Asia. Oh, sorry, but the slides seem to be changing automatically before I'm ready for them, so I may have to occasionally flip back like that. These are Trogontherii-like mammoth teeth. You can see the high crown. They've got 20 to 22 plates. They're really like Trogontherii, but they are there much earlier than we have them in Europe. The earliest ones that were found were the ones excavated by André Scher in northern Siberia. You can see on the left there in the early Illyrian, which are around 1 million years old. And then in 2003, Chinese colleagues announced these remains from the Majuangu site, which are as much as 1.7 million years old. So while Meridionalis was still dominant in Europe, we have got early Trogontherii in Eastern Asia. So we have to suppose that around 2 million years ago, there was something like an allopatric speciation event, because we know that there was Meridionalis in that region before. So it looks like an eastern population of Meridionalis split off and evolved into the Trogontherii species, rather as shown in the map there. And then just to recap what that change represented, Meridionalis, which with its relatively lower skull and longer skull and low crown teeth with typically 10 to 14 plates in the M3, but Trogontherii with a higher skull and a higher tooth. Of course, those two are connected anatomically, and a greater number of plates in the molar. And as corroborated by dental microwear analysis, the Meridionalis kind of molar more browse adapted, the Trogontherii one more graze adapted. I would say really it's a transition from browse dominated mixed feeding to graze dominated mixed feeding. So, you know, it is a transition, but it's not an absolute transition from one to the other. And there are the artist reconstructions of those two species. And it kind of makes sense that this would have happened, because we know from the paleoenvironmental evidence that already by about 2 million years ago, early in the Quaternary, in China, the climate deteriorated, became more seasonal, and there was the opening up of grasslands, which presumably is what selected for the evolution of the Trogontherii type. And I've called that the biggest single step in mammoth evolution. And then, as we know from André Scher's remains from northern Siberia, sometime by about 1.5, maybe 1.25 million years ago, this species had spread up beyond the Arctic Circle into northern Siberia, northeast Siberia. So that was the situation at about 1 million years ago, with the continent divided between those two species. So then when we come back to that question of what were the mammoths that first went into North America, which one of these was it? We've now got a choice, basically. And previously, many mammoth remains from North America were taken to be something like Mammuthus meridionalis, early mammoth remains from early sites, from early Pleistocene sites in the US, in Canada, and in Mexico, were taken to be meridionalis in support of what I call the standard model, which is that was the first species that went across into North America. But I noticed when I studied all these remains that these supposed meridionalis forms, they all turn out to be very deeply worn lower M3s. And I think this is no coincidence, because if you studied elephant teeth or mammoth teeth, you'll know that in the lower molars and in the M3s in particular, the plates splay out towards the base of the crown. So that if you have a tooth that's in relatively early wear, the plates are quite close together and you get the full plate count. And so it looks relatively advanced. Once the animal gets very low in wear towards the bottom of that picture, the plates are more widely spaced, and of course it will have lost plates at the front due to the natural erosion of the tooth. So it tends to look more primitive, like a meridionalis, but that's actually deceptive. And if we look at the early Pleistocene mammoth sample from North America, which is the best preserved, where we have a whole sample of molars which are in relatively early wear, very well preserved. This is from the Lazy Shell Pit in Florida, which is more than 1 million years old. But these teeth show what real early Pleistocene North American mammoths look like. They're really quite advanced. They've got the high crowns, they've got more than 20 plates, they've got a relatively advanced mandible. To all intents and purposes, they are virtually indistinguishable from the late Pleistocene Mammuthus columbi, and I'll come back to that later. I have not, in all my travels around North American museums, ever found a molar in North America that looks like this. This is a classic European Mammuthus meridionalis. Thanks to Ebru for this photo. I believe she's listening in on this. This kind of very low crown molar with only 12 plates, which is, where you've really got meridionalis. I've yet to see a specimen like that from North America. Maybe one will turn up and we'll have to revise the story, but that's the situation as it stands. Conversely, when we look at North American mammoths of different ages through the Pleistocene, they're incredibly similar metrically and in appearance to classic Eurasian Trogontherii, as you can see in those photos. So I think all the evidence points to Trogontherii being the ancestor of the North American columbi for all the reasons that I've listed at the bottom. Nothing like meridionalis has ever been found in North America. North American mammoths are very similar to Eurasian Trogontherii. Trogontherii was the first species of mammoth that extended that far north into Beringia. So it was, if you like, it was in the waiting room for the transit across Beringia into North America. I just want us to then think about the dispersion of Trogontherii westwards. We looked at the eastern dispersion. What about the western dispersion into Europe? And when Trogontherii started to disperse into Europe from about 1 million years ago, it was entering the territory still occupied by the meridionalis species, something like that. About 750,000 years ago, we do have some sites in Europe, in Britain, in Germany, and maybe elsewhere, where you can see temporarily these two species coexisting. But then by the end of the early Middle Pleistocene, meridionalis had gone, and the picture across the world is very simple. We've got Trogontherii right across mid and high latitudes of Eurasia, with columbi south of the ice in North America. And then comes along Mammuthus primigenius, the woolly mammoth. And once again, it's the remains that André Scher excavated from somewhat later deposits in northeast Siberia that have produced the earliest known fossils identifiable as woolly mammoth. These have increased the plate count to up to 24 plates, thinner enamel, the whole woolly mammoth package. And these from the late Illyrian are between about 0.6 and 0.8 million years ago. As such, they're the earliest woolly mammoths from anywhere. And I think from a biogeographic point of view, it's the same story again that we had when Trogontherii speciated from Meridionalis. Now we've got presumably a population of Trogontherii in northeast Siberia. Again, it makes ecological sense because obviously this was an area which, as the ice ages progressed, had very harsh climatic and vegetational conditions, provided the further selective pressure transition into the woolly mammoth species. And then through the Middle Pleistocene, Primigenius spread eastwards into North America, south and west into Eurasia, until by the late Pleistocene, this was the situation: woolly mammoths everywhere in the northern realm, Columbia in North America, in the southern part of North America. So the two species kind of dividing the continent between them, but we'll have a bit more to say about that in a moment. I just want to scroll back a few slides to think in a little more detail about this spread of Primigenius into Trogontherii territory in Europe, because it's interesting to consider in more detail how that kind of transition takes place. And here I've got a slightly complex looking graph, but actually all it's showing is the plate number of the third molar. You can see it at the bottom, ranging from about 16 to 28, through a whole series of samples, mostly from Europe. Those are the grey ones, and a couple of important ones from Siberia. Those are the blue ones, going up through time. And from about 600 to 200,000 years ago, shown by the green shape there, there's essentially stasis. There's no real change through the Trogontherii sequence in Europe. But then, as we've explained in the earlier slides, Primigenius from Siberia with a higher plate count invaded that Trogontherii territory. And the result of that in the middle to late Pleistocene of Europe is not a clean transition. They're not just all like Siberian Primigenius. What we've got are a series of samples that actually span the morphological range of the two originating taxa, if you like. Some of those samples have a very wide spread. Some of them are more on the Primigenius side. Some of them more like Trogontherii. What does that actually mean? I'll show you what I think it means in a map in a moment. But just interesting to notice that even by the latest Pleistocene in Siberia, we still have what you might call clean Primigenius, just like the original Siberian population. I think that's because northeast Siberia was the place where Primigenius originated. So the whole Trogontherii population in that region transformed into Primigenius, whereas in Europe, you've got it migrating into an area with Trogontherii in it. And so there was some more complex kind of interaction, including probably hybridization. So if we put that into maps, we started with the Middle Pleistocene broad range of Trogontherii. We had the origin of Primigenius in northeast Siberia, like that. Then we had the spread into Europe. What I think the end product is, if we transform that graph into kind of map form, this is purely schematic. I'm not saying that these are the actual outlines of populations. This is just schematic. But I think we had some populations that are more purely Primigenius. There are some that retained a more Trogontherii morphotype. And then there are others which may well be hybridized, which have something intermediate. The black arrows suggest gene flow between them. And of course, these probably would have moved around with climatic cycles. So you have a very complicated situation, which in modern biology is known as a metapopulation structure. Right, well, now we're going to move on to the DNA evidence. And I'm basing this mainly on the paper that we published a couple of years ago, the work really done by colleagues in Stockholm, Ton van der Valk and Patricia Pesnarova in Lova Darlin's lab. And I basically provided the morphological angle on it. So for this study, we deliberately chose the very specimens that Andrei Scher had excavated from northeast Siberia, which I showed you earlier represent some of the earliest Trogontherii and some of the earliest Primigenius. These specimens are named after the localities they came from. From the early Pleistocene, the early Trogontherii type, we've got these two specimens from Krestovka and Adicha, and then from the earliest Primigenius sample in the Middle Pleistocene, we've got that specimen from a site called Chukotka. And this diagram shows in simple form the basic findings of the molecular study. You've essentially got a very simple tree there, and you can see where those three specimens sit on it. And at the top of the graph, you've got the late Pleistocene Columbi on the right and the late Pleistocene Primigenius from Europe and North America on the left. And I'm going to go through this graph showing in sequence the main points that this illustrates. So first of all, the first thing it showed was that the early Primigenius in Siberia is a direct descendant of the early Trogontherii in that area. So that I was very pleased with that, because that confirms what we thought from the morphology, that northeast Siberia was probably the earliest place that Primigenius arose, and that it arose from the indigenous Trogontherii in that area, and the molecular evidence seems to corroborate that. Second point is that we can then trace that lineage further to late Pleistocene woolly mammoths in both Europe and North America, and that further corroborates the idea that we had, that I showed you on the map, that early Siberian Primigenius population then spread east and west to fill out the whole of the northern realm around the globe. So far so good. A finding from the molecular data which we had not suspected on the morphology is that the Trogontherii mammoths from northeast Siberia turned out to comprise two very divergent genetic lineages. Now, morphologically, you can't really tell them apart. Don't pay too much attention to those particular photos. Those are just the individuals we sampled for the DNA. It's a large sample of teeth, all of which I measured. You can't really distinguish these two genotypes, but genetically they're very distinct, and you can see that there. They probably diverged from each other about 2 million years ago. And then the final very fascinating finding was that the other genetic lineage among the Siberian Trogontherii, the Krestovka one, looks to be an ancestor of North American Columbi. So that's very nice as well, because we also assumed, if you remember, that North American Columbi came from the northern Siberian Trogontherii crossing the Bering Strait probably about 1.5 million years ago. So that's all great. That all kind of fits the morphology very nicely. There we go. So that summarizes what I just said. And then the genetic data had another very big surprise for us. And that is that the late Pleistocene Columbi of North America is only 50% genetically derived from the Siberian Trogontherii. It had another 50% from early Primigenius. So this must have been a second wave of migration into North America, which is calibrated from the molecular data as Middle Pleistocene, from the early Primigenius of Siberia, came into North America, and something like this must have happened because the late Pleistocene Columbi is a hybrid between those two source populations. And so this very nice illustration suggests what happened when those two populations met. And are we to say then really that the origin of the late Pleistocene Columbia in North America is really a result of that meeting of those two populations? So we could actually call it a hybrid speciation event. So the classic Mammoths columbi, is it a hybrid species? Should we call it a hybrid species? I'm going to focus a bit more on that North American lineage. And in the next slide, you'll see essentially a close-up of what I have marked with that orange dashed line there. So here we go. Here's that North American lineage. And this hybridization finding is, of course, fascinating, but it does create some problems for us as paleontologists. First of all, that genetic data implies that there was a very early Middle Pleistocene migration of early woolly mammoths into North America. But in fact, we have no fossil trace of woolly mammoth coming into North America that early. So is it that it was a very small population initially, and that's why we haven't found any fossil trace of it? Will we find them in due course? Are some of the undated primigenous fossils from Alaska, for example, are they perhaps Middle Pleistocene? And we don't know because they're just not dated. That's a kind of standing puzzle. Secondly, there's a kind of taxonomic problem that this faces us with, because if we are to say that the Columbi species, which of course was named on the basis of late Pleistocene fossils, if we're to say that it actually originated at that speciation event, it means that the early mammoths in North America, the ones that first came from the Siberian population, need to be named as a separate species. I called it Mammoths XXX, like that. Okay, so at that hybridization event, there was, you know, essentially one species became another by hybridization. But there's a problem with that, because there's no real morphological distinction between the early Pleistocene, Middle Pleistocene, and late Pleistocene mammoths in North America. This is a plot of two of the key parameters, taxonomic parameters in mammoth molars. This is the lamellar length, it's similar to lamellar frequency, and the enamel thickness. And what you can see here is the yellow are early Pleistocene North American, turquoise are Middle Pleistocene, and purple are late Pleistocene Columbi. There's essentially no difference between them. And the fossil that I've shown at bottom left is actually one of those early Pleistocene 1.3 million year old fossils from the Florida site that I showed you on a slide earlier, which I said at the time, you could hardly tell it apart from late Pleistocene Columbi. So that gives us a problem, because if we want to call the early Pleistocene mammoths a new species, Mammoths X, how do you define it? Because there's no morphological distinction, at least not that we found so far. Oops, and I admit we've only so far looked at teeth, but on that basis, there's morphological stasis, so there's no morphological basis on which to define a separate species from Columbi. And so far, we don't have any ancient DNA from those early North American mammoths. We're working on that. So, what's the differential diagnosis, and how would you choose a type specimen? And further to that, the date at which this supposed early primigenous migration into North America happened is not based on fossils, because I've told you there are none. It's based on molecular calibration, and that actually gives quite a wide range of possibility. We don't know exactly when that hybrid event occurred. So you couldn't really split those two species on the basis of a chronological point. I don't think that basing taxonomy in the fossil record on pure chronology is a good idea anyway. But you couldn't say, well, we know that the hybridization was at exactly 500,000 years ago, so anything before that is one species and everything after that is another species. And even apart from that, of course, many fossils are not dated to, or most fossils actually are not dated to that degree of accuracy that you could use that method anyway. So what do we do? I mean, I basically would suggest that for the time being, we don't do anything. I think actually for the reasons I've just explained, I think we should continue to call all North American mammoths columbi. They are morphologically essentially the same. I say that with a proviso that we only know about teeth. Maybe in the skulls or in the postcranial skeleton, there might be clear markers that would help distinguish that, but so far that's not been discovered. So I think we should keep it as one species, which is a morphotype, with this very interesting new knowledge about the later ones having a big input of primigenious genes, as of course a very important evolutionary and genetic piece of information. Or if we want to be a bit more cautious about it, we could add a CF into those early ones. One final surprise that the molecular data gave us is that there was a second hybridization event into North America. Now, this happened in the late Pleistocene, or maybe about 100,000 years ago, something like that, and where another 10% of DNA came in from primigenius to columbi. So actually late Pleistocene columbi ends up with about 40% from its original Siberian Trogontherii ancestor, and in total about 60% of primigenius from successive ways of migration across the Bering Strait. And of course, this is much less of a surprise because suddenly in the late Pleistocene, we've got lots of primigenius fossils in North America, so that doesn't present a problem like the earlier migration did. And what's more, now we start to see a morphological consequence in that I've added to the same graph late Pleistocene fossils from North America. And we've got the purple ones, which have been considered to be of columbi type, but then we've got the primigenius type, which are the blue circles. And now we've got a complete spread because the purple ones on the other side of the dashed line are still late Pleistocene. So late Pleistocene North America covers the entire graph because you've got the purple ones, which have traditionally been called columbi, overlapping in the middle with the blue ones, which are the incoming primigenius, and the ones in the middle that have been called Jeffersonian type Mammuthus columbi may well be ones with a greater input of primigenius genes into the North American columbi population. That's rather speculative at the moment. So whereas oddly, the big 40% input of primigenius genes in the middle Pleistocene didn't seem to have any morphological effect. The late Pleistocene one did. Maybe that's because by the late Pleistocene, the environmental conditions were more severe, and so the primigenius morphology continued to be selected for in North America, whereas earlier the columbi morphology continued to be selected for even with the input of primigenius genes. That is all entirely speculative. And just to wrap up, I showed you this slide earlier, which was the conclusion as to roughly what late Pleistocene mammoths in Eurasia kind of look like. I think on the basis of what we've seen in terms of what's going on in North America, so we had successive migrations into North America. You've got the green sitting early North American type, Trogontherii-like type at the bottom there. In comes primigenius. And I think that the end product is actually quite similar to Europe, because what I tried to show in that last diagram was that in late Pleistocene North America, you have some populations that are more like columbi. You have some populations more in the north that are more like primigenius. And you've got others which kind of span the two, or they are intermediate. They may have more of that late hybridization within them. And so the whole thing is a complex metapopulation. And of course, this makes putting things into boxes really quite tricky. Classic Linnaean taxonomy is a bit difficult. I mean, this is wonderful from a point of view of understanding how evolution works. It's not so great for curators who want everything to have a nice name. But that's the way evolution works. So Thanks very much for listening. And just to say that a paper which Lova and myself have written is literally highly imminent. So most of what I've told you about today is in this paper, and it's coming out within certainly weeks, possibly even days, in the journal Paleontologia Electronica. It'll be open access. So take a look at that, and I'm very happy to answer your questions if I can.
Thank you very much, Adrian. Wonderful tour through all the Mammoths evolution. Certainly for us in Mexico is great to learn all these latest outcomes from your research. So far, we don't have questions on the YouTube, but certainly they probably will come. Probably one, and well, Dimila says her greetings to you.
Right.
Anyway, one question I will have is, where Mammutus exilis extends, did it actually evolve out of Columbi or Columbia?
Did you say Mammuthus exilis?
Yes.
Yes. Oh, yes. I think, I mean, that's a good question, because now we've got this more complex situation in North America. I mean, because they are pretty far south, you know, they obviously came from California. I think they came from middle to late Pleistocene Columbia.
Okay.
Yeah, and I would assume post-dating that hybridization, so they probably carry some of the primigenius genes. But that's a very interesting question. I don't know that there's been any genetic data from exilis.
No, so
yet.
far as I have not yet. In fact, the other population that from time to time has been also questionable is the one from Wrangler Island, but
Yeah.
I don't know if it also stands still as part of the Trogontherii or.
Oh no, Wrangell is part of the Mammuthus primigenius.
Okay.
I mean, yeah, it's one of the primigenius clades that got there pretty late. Yeah, and it's, you know, there probably is some kind of decline in the degree of introgression of primigenius into the endemic population. So your mammoths in Mexico, you could predict, might have the highest percentage of the original population in, you know, the primigenius genes, you know, penetrated down the continent in some sense. So we're very keen to get DNA, maybe from your samples. I don't know if anyone has tried yet.
Well, currently there's a Mexican team at Querétaro trying ancient DNA.
Great.
Guided by Dr. Federico Sanchez, who has been working along with the Stockholm people. So
great.
Anyway,
yeah, that's
also Ebru sends
hi.
regards. Anyway, so you are very
I'm glad
popular.
all my friends are watching. That's great. Yeah.
Anyway, so that's the other thing I wanted to mention. You may remember a few years ago, Emil Karpinski and his colleagues, they published on this mammoth genetic data that it showed that probably some mammoth Americanum went down to even to central Mexico, and probably some population went back. Will you expect that with Mammurus?
So populations of which?
In that case was Mammoth Americanum.
Oh, yes, mastodon. Yeah.
Yes.
That they. Can you repeat the question, Joachim?
If you recall, they mentioned the possibility that there were the migration of this Mammoth American population from northern United States, actually Canada, into Mexico, and probably some of those coming back to the north.
Yeah.
Do you think, would you expect that from Mammurus?
I think these processes are very complicated, and the thing with the genetic data is it doesn't necessarily need movement of animals, because genes can float through a population. So I think it's very difficult to reconstruct. We need morphological markers.
Okay.
It's tricky. Of course, it's entirely possible, but I don't think we've got that level of resolution yet.
And in fact, you may remember also that paper by Dudley et al.
Yeah.
That they named a new mammoth species,
Oh,
Pleistocene
yeah.
species.
Well, there's the Mammut pacificum.
Yes,
in,
exactly.
Yeah, well, there they believed that they had some dental and postcranial characters which enabled them
Yes.
to do that. And so far we don't have that for mammoth, but I think with more study, I hope we will be able to refine this better. Yeah.
Good. And I really like that you also do a lot of encourage the morphology data, the diagnostic morphological data, because and right away Marisol Montellano is sending her regards. She has a question, but she herself has been pointing out to students that they should not only rely on metric differences or other, but they should look for characters. Let me read to you the question that Marisol has. Would you expect that the M. columbi from Mexico is more pure?
I think it's quite likely, yes, because the Primigenius came in from the north and spread south, and there would have been gene flow to the south. But if anywhere would preserve more purely, if you put it that way, the original Trogontherii from Siberia, from the early Pleistocene, it would be in the south. So yes, and I think this is why it will be very exciting to get some DNA from the Mexican mammoths. I would say it's a good hypothesis.
Okay.
We need to test it.
And in fact, she complete the question mentioning that get more isolated from the northern population. So
I'm not sure I'm not sure it was isolated, because we have mammoths in the southern US, Texas,
Yes.
Florida, and so on. So I don't think we need to think of it as isolated. So I think there would have been some gene flow. I will be surprised if it is 100% pure, put it that way. But that's the question. If it turned out to be, identical to the original Siberian Trogontherii with no primigenius input, that would be evidence of isolation. But from a purely geographic point of view, I would have thought it's more likely to be continuous, but with less introgression as you go further south. Yeah.
And well, I have myself another question. Based on this different movement of the European populations, do you think from the Cape art that had appeared in mostly Western Europe, Central Europe, all these wonderful drawings in caves, do you expect or do you think it can be separated different species of mammutos? I know it's quite difficult, but it's just wondering.
I think that would be pretty difficult, because there's a lot of different artists are representing them in different ways anyway. No, I don't. I think that would be unlikely. But now you've mentioned it, someone will probably try.
Okay.
One thing to mention there, actually, which is relevant to that point, is that one of the other things that our genetic study showed, which I didn't mention in my talk, is that already in the Trogontherii, in the early Pleistocene, the earliest Trogontherii, it already has many of the functional genes that Primigenius had as an adaptation to cold climate. So the functional genes in the early Trogontherii suggest it already had a thick coat, it already had a thick fat layer, and so on. So I think, so I think the appearance of the animal was not actually so different. Maybe the fur was not so thick as in Primigenius, and we know that they were larger actually in size. So I think the external anatomy was probably not so different. we have these good differences in the teeth. So again, I think it makes it a bit less likely that we'd pick it up in the cave art.
Mhm.
Yeah, this is all very
Eh,
speculative.
Marisol keeps sending some interesting question. Would you expect a geographic variation between the Colombian mammoth from North America and those from Central America?
It's possible. Yeah. I think we don't know much about the Central American mammoths yet, but yeah, I mean, you might expect that if they're at the edge of their range, that is the southernmost edge of their range, that the populations might be more fragmented because the suitable habitat would be not continuous. If you're starting to get into a zone where there's a lot of forest, for example, because you're really subtropical or tropical, then the areas that the mammoths could inhabit might be more fragmented, which could give more opportunity for isolation. So I think, again, I would call that an interesting hypothesis, which I think the only way we will test that is if we get DNA. And of course, the problem is that the more tropical you get, the poorer the DNA preservation normally. But these lab guys are getting pretty smart at that. So
I know.
we must hope, yeah.
Well, actually, from what we have, and my colleague Federico has been checking with all material, and the most recent one from this big excavation at the International Airport in northern Mexico City, seems that those fresher samples are better than the old ones, and certainly you can understand that.
Good. Yeah, great.
I think we don't have any further question. Eduardo, is there anything else? Okay, well, I guess we can wrap up here. We really appreciate your help. I think has been wonderful, and as we had mentioned, this will be staying in the channel of the coordination in YouTube and also on the seminar webpage. So I'm sure that much people will be checking on that, mostly because of the time schedule of those for you in England. Probably some people is already having dinner or going to bed anyway. We really appreciate your support, Adrian. As I mentioned, having very supportive, and I really will be proud that in your third edition of your mammoth food, you mentioned tequila. That is wonderful that you consider our site. But anyway, thank you very much for your support, and look forward to meet you next year here in Mexico City.
Yeah.
I don't know. Thank you very much.
Okay.
Much, Adrian.
Bye.
Well, thank you. We will just have some advertisement for the people on the YouTube page. Eduardo.
Ok, gracias. Bueno, después de esta excelente charla, quiero comentarles que les voy a compartir la pantalla. ¿Dónde está? Acá está. Y ya mañana vamos a tener disponible el libro de resúmenes de este simposio virtual, y va a contener los resúmenes y los accesos a los videos que están en la página de la Coordinación Nacional de Antropología. Entonces, bueno, pues ese es uno de los avisos que queríamos darles. Ya mañana estará disponible. Va a estar colgado en la página del Seminario de Relaciones Hombre-Fauna, y pues se va también a repartir por correo electrónico a través eh, el correo del noveno Congreso Internacional de Mamuts y sus relativos. Entonces, pues les pedimos para que estén atentos y bueno, pues para que tengan este libro de resúmenes. Y el otro anuncio que queríamos hacerles es que ya vamos a continuar con las sesiones normales, y el 24 de diciembre, ándale, 24 de octubre vamos a tener la siguiente charla, que es a cargo de Nicolás del Sol, que ahorita está en la Universidad de Laval en Canadá. Y se va a tratar de, bueno, el título es Vacas, Genes y Vaqueros, la paleogenética y la historia ganadera en el México colonial. Tomó unas muestras de ganado vacuno y bueno, sobre esto se ha hecho todo un trabajo muy interesante. Entonces, bueno, cambiamos de tema, pero sigue siendo el tema de interacciones humano-fauna. Y bueno, pues la idea es que nos veamos entonces el 24 y después ya nada más nos quedará la sesión de noviembre, que va a ser sobre pintura rupestre. Ya también les vamos a dar el anuncio formal. Ese va a estar a cargo de Bean Meléndez, que está de la ENA. Y bueno, ya daremos los anuncios formales a través del correo y de los medios usuales que utilizamos. Entonces, pues nada, les agradecemos mucho la atención. Gracias, Joaquín. Y pues ya te dejo para que despidas formalmente el simposio, para que le des cierre formal al Simposio Internacional Virtual que se organizó.
Thank you, Eduardo, and thank you everybody for assisting to these presentations. As mentioned by Eduardo, they will be held in two web pages, mostly the one from the Seminario Relaciones, Hombre-Fauna, and you can look at them anytime. We have 6 wonderful talks, and I'm sure this will encourage and hope encourage all of you to try to come to Mexico City next year. Within a month, we hope to send our call for the abstracts and for this forthcoming meeting. So we will send all those on the web page. And if you want, on the web on the group of the Congress, just send us a message to the seminar page, and we will send you the calls. And well, thank you again to all the people from the Coordinación Nacional de Antropología here for their help in putting this seminar on the web as well on YouTube, and hope to see all of you in the forthcoming seminars of the series, and also next year here in Mexico City in September 2025. Thank you very much, all of you, and bye-bye now.
Instituto Nacional de Antropología e Historia.
Secretaría de Cultura.
Gobierno de México.